Today topic of discussion is Pox Viridae. Morphology. It is a brick-shaped virus. It have genetic material, double stranded DNA, enveloped virus. Symmetry is complex. Replication takes place in cytoplasm. So morphology of Pox virus: brick shape, double stranded DNA, enveloped, complex, replicate in the cytoplasm. This virus, brick shape virus, see the diagram. This virus look like a brick shape virus, rectangular. All DNA virus are double stranded except parvo. So this one is double stranded DNA. Parvo only single stranded. It is a enveloped virus. This one having complex symmetry. Rest all DNA virus having isosahedral. So Pox virus only having complex symmetry. And it replicates in the cytoplasm. Between capsid and envelope. Between capsid and envelope, two lateral bodies presented. Shape of capsid is dumbbell shape. This one is the dumbbell shape. It is a typical diagram. No other virus have this diagram. It is one of that family of Pox Viridae. So nucleic acid contained with a dumbbell shaped core on either side of the DNA core is a lens shaped structure. It is called as lateral body. It is the largest DNA virus. Overall also largest DNA virus. So we can see by using light microscope. This is all about morphology of Pox Viridae. Classification. How many Pox Viridae family members are? Totally 9. Totally 9. Now Pox Viridae family, among 9, 3 will causes diseases to human. Variola, Vaccinia and Molluscum contagiosum. So Variola, Vaccinia, Molluscum, contagiosum. These 3 are causing diseases to human. So started with Variola. It is the most important. Second one is Vaccinia and third one is Molluscum contagiosum. Apart from monkeypox, initially causes diseases in monkeys and squirrel, but nowadays it transmitted to human also. So monkeypox, cowpox, Warf-pox, Tanapox, Yapha-pox causing diseases to human. Human only Variola, Vaccinia and Molluscum contagiosum causing diseases. So important Pox viruses are Variola. It is causing agent of smallpox. Then Vaccinia, artificial virus which was used for smallpox vaccine. Third one is Molluscum contagiosum. Important of this family is Variola, which is a causative agent of smallpox. Smallpox is the name of the disease. The virus causing smallpox, that organism is the Variola. So organism name Variola, causing diseases name is called smallpox. That is the first virus. Second virus, Vaccinia. It is not a natural virus. So this one is not a natural virus. It is the artificial virus which we have. Human have prepared from the Variola to prepare the vaccine of that Variola. Less attenuated virus from of the variola is known as Vaccinia. Why this one called Vaccinia? Because it is used for producing the vaccine. And the third one is Molluscum contagiosum. So we will discuss these three in detail. Started with the first one, Variola. The root, how does it enter human body? Main route is inhalation. By inhalation only this virus will enter, which causing disease which is known as smallpox. But nowadays there is not even a single case of smallpox in the world. It is already eradicated. But you can see it look like chicken pox. Rashes only common. It is enter human body via inhalation. Then it enter, after that it will go to the nose. Then it will go to the trachea. It will go to the bronchi. Then it will reach the lungs. From lungs it will go in lymphatic and blood. From the blood it is having a strong affinity for the dermal tissue for the skin. It will go in the skin and form small vacuoles. The vacuoles are known as Pox. Okay, this one Pox. The fluid filled vesicles are known as Pox. So here Pox virus producing Pox. See the spelling difference. Pox is the name of the virus. This Pox, P-O-C-K-S, this one is the name of that vesicles which is produced by it. Name of that vesicle, this name of that virus. So classical Pox are produced. So this is the pathogenesis. Through inhalation it will enter, then it will reach to respiratory tract, then disseminated via lymphatic. This virus will enter to blood, viremia. Then it will infect all dermal tissue and internal organ. Then it will causing Pox, P-O-C-K-S. Pox virus produced Pox. Name the disease. The name of the disease is smallpox. So the smallpox rashes began from that mouth. It started from mouth part. Then it is distributed throughout the body. It is going away from the center mouth. That's where the distribution. This is known as centrifugal in nature. This one is the centrifugal in nature. Away from that center. So the rashes benign from mouth, then spread throughout the body within 24 hours. All rashes first. All will be papule, then all will be pustule, then all will develop finally into vesicles. Finally scar will be formed. Completely body, all the rashes will be same stage. All the okay, all the rashes will be same stage. All will be papule, if all, then all will be pustule, then all will be vesicle, then all will be scar. For example, it is starting from papules. Every one will be papule. If started from papule, it will then all develop into pustule. Then it will develop into vesicles. Then all develop into scar. So The rashes is synchronous. That rashes is synchronous. That is in same stage only. All are same stage. But chickenpox occur in various stage. But here all are same stage. Everywhere same stage. So 5 to 17 days incubation period, prodrome of flu-like illness for 2 to 4 days, then prodrome followed by rashes, this one benign in the mouth, then spread into that face, arm, legs, hands, feet, can cover entire body within 24 hours. All vesicles are the same stage of development, so it have that name synchronous in nature, synchronous rash. Then smallpox, it having that multiple compartment. We first that lesion, it having multiple compartment is presented. No inflammation around the vesicles. Here vesicles is presented mean surrounded, no inflammation. But chickenpox having inflammation, it will affect form and soles. Then slow evaluation, scar from after 10 to 14 days. Then world has eradicated only one disease till date, smallpox, declared eradication on 8th May 1980. So first eradicated diseases in the world, smallpox only, declared in 8th May 1980. Next, targeted tissues for eradication globally, polio, measles, guinea worm and diphtheria. List of eradicated disease list. World is declared 8th May 1980 free of smallpox. After that, still 2023 no cases. Last indigenous case of smallpox in India 17th May 1975. Last importation case of smallpox in India 24th May 1975. India declared smallpox free in April 1977. Last case of smallpox globally 26th October 1977. Then actual last case of smallpox laboratory accident 1978. Then globally eradication of smallpox 8th May 1980. The smallpox virus has not been completely destroyed from that environment, but it is presented in laboratory US and Russian laboratories. Still now that virus is present. Then treatment, there is no treatment, prevention, vaccines available. At that time, not now in a days, nowadays not in a single case also. Next, Vaccinia virus. It is not a natural virus. It is an artificial. Humans have produced this virus from Variola only by changing one antigen. How they prepared? By changing one antigen. So if we change one antigen, the Variola is converted into Vaccinia, and this is non-pathogenic. It is used for vaccine. So we convert variola into Vaccinia by changing one antigen. So by changing one antigen, variola is converted into Vaccinia. So we are making it less pathogenic but more immunogenic. This one less pathogenic, more immunogenic. So we can use this virus for producing vaccine. So that all about it. Third one is, this one is the difference between variola and Vaccinia virus. This one is the natural virus. This one is the artificial virus used for vaccine preparation. These are the main point. So main different, this one is causing smallpox, this one is the artificial virus used for vaccination. Then it have elementary bodies, name is called as patient's body. It have elementary bodies, name is called as cuniary bodies. Cuniaris bodies. These are the main different. So third virus name is called as Molluscum contagiosum. The only virus which cannot be grown in egg and it causing disease is known as Molluscum contagiosum diseases. It may rashy. The center in this rash, the center part it having depressions. In the center there is a depression in the rash. This is known as amplificated rash. This one name is called as amplificated rash. Next, laboratory diagnosis. Specimen we will collected vesicular fluid, 3 type of virus, Variola, Vaccinia and Molluscum contagiosum. We know 3 type of virus. By using syringe from that vesicles, whether it is smallpox, whether it is Molluscum contagiosum, after that two things of vesicle fluid. First one is vesicular fluid directly make a slide and do microscope. In the microscope you can see that light microscope. The light microscope also this virus you can see inclusion bodies. What are the name of inclusion bodies? In the case of variola, the inclusion bodies name is called as basin bodies. Basin bodies. In Vaccinia virus, cuninary bodies. Cuninary bodies. Then Molluscum contagiosum, it having Henderson, Patterson's bodies. This is the scientist name. So 3 type of inclusion bodies. By using microscope we can find out. Variola means basin body. Vaccinia means cuninary bodies. Molluscum contagiosum means Henderson, Patterson bodies. The scientist who discovered these bodies. The next way of diagnosis that embryonated egg, hen egg. Here we will take egg inoculated that virus. 4 site of inoculation. 4th site of inoculation. One is chorioallantoic, that outer covering, chorioallantoic membrane. Second one is amniotic cavity. Third one is allantoic cavity. Fourth one is yolk sac. See that tip of that syringe is We take specimen in the syringe and inoculating into 4 layers. Here we are not using the 4 layers. First layer CAM, then Korean allotonic membrane only we are using. That outermost layer, that is chorioallotonic membrane. Take vesicular fluid in a syringe and inoculated outermost layer. After that, okay, here that outermost layer we will inoculated. After that, keep this next 48 hours, 48 hours for the inoculation. Then after that, so if the virus is presented in the fluid, we will get replicate in this that Korean allotonic membrane. After 48 hours, hatch the egg, take that membrane out. If the fluid containing vesicles, it will form small lesion on the membrane. These lesions are known as Pox. Pox, Pox. So it producing Pox. Only Molluscum contagiosum cannot be grown in egg. Remaining Variola virus can grow. Remaining all Pox viruses can grow in egg inoculation. Thanks for watching.